Good evening, all. I invite Dr. Kunjal Patel today, who's a molecular pathologist at the Newburgh Center for Genomic Division, to introduce the first speaker for the session and start the proceedings. Over to you, sir.
Hello, good evening, everyone. Myself, Dr. Kunjal Patel, and today's speaker is Dr. Vidya. Dr. Vidya is a principal scientist at 4baseCare, and she has an experience of more than 15 years. Apart from that, she has more than 30 publications done, and she has been previously working with affiliated with Genome as well as HCG cancer centers, and her primary areas of interest is minimal residual disease in tumor and leukemias. Apart from this, she has validated the first liquid biopsy project in India, which include solid tumors as well as other neoplasms. So, Dr. Vidya, stage to you, please. Thank you.
Thank you so much, Dr. Kunjal, for this introduction. Without much delay, I will start my presentation. I'll share my screen here. Today, the topic of discussion that has been where I have been asked to speak is about complex genomic biomarkers, MSI and TMB. I'm just going to give some historical information in the interest of students. Cancer is essentially a phenotypic transformation of a healthy cell into a malignant cell by acquiring mutations, which are essentially the somatic mutations. And this was the concept which was early on endorsed by two very famous scientists, Hanahan and Weinberg. They published seminal articles in 2000, 2011, and 2022. What it essentially says is it involves multiple mechanistic pathways, and you need to look at how a normal cell transforms to a malignant cell by incorporating mutations into its own genome. Now, I am just in the interest of time, I'll be going fast, but please stop me if you have any questions to ask. I have put up here way back in the year 2000. We knew that there were 6 hallmarks. Then in 2011, there were 10 hallmarks. In the the recent 2022 has many more, but it's far more easier for us to understand the 10 picture. So that's the reason I have put up here. So essentially what it does is, we all know about proliferation, angiogenesis, and then loss of the apoptotic machinery to work efficiently. And then it also had several other mechanisms by which it acquires a malignant transformation. After acquiring, it has to sustain. To sustain, it develops several other mechanistic pathways, among which one such pathway is to evading the immune response. It tries to evade the immune response. So when a healthy cell gets mutated into a cancer cell, what is essentially happening? The cell becomes a foreigner. So what it should do essentially, it has to be recognized by the immune cells, at T cell mediated immunity, and then the host cell will kill the cancer cell as if it is a foreigner by using NK cells and T cells. However, what the cancer cell does is, it evades this recognition by and bypasses this immunological killing by activating some of the inhibitory pathways. These are known as the immune checkpoints. And this is what the cancer cell smartly utilizes to survive, even if the host immune system is very good. Now, how are we going to ensure that we block this lock and key which the cancer cell has already built? By interacting with these immune checkpoints, it essentially plugs the immune checkpoint, and then it stops its function of doing this job of NK cells and T cells killing the cancer cell. So subsequently, we had a targeted therapy. Who are these markers? Now I have a picture here. So this is how it is a presentation. And then essentially the mutation is presented as a new antigen, which is recognized on top of the MHC receptor. The T cell recognizes and kills it. but there are some neighboring receptors like CTLA4, PDL1, and PD1, which the cancer cell tries to block and ensure that this T cell does not recognize the tumor cell as a foreigner. Now, what we are doing as humans right now is, once we understood this process, we want to block these receptors so that the T cell recognizes the tumor cell as a foreigner and then it tries to kill the cancer cell. So, what are the landmark achievements that we had? Very first achievement was back in 2011, ipilimumab, which was an anti-CTLA4 antibody, and it has shown an excellent response with melanoma. Then we had PD-1 inhibitors, which are nivolumab and pembrolizumab, which was approved by FDA for these cancers, and so on and so forth. Now we have identified a pathway, and we know that you can block this pathway by trying to outsmart the cancer cell by blocking these receptors CTLA4, PD1, and PDL1. So now, who is the patient or which tumor is classified as an eligible tumor for immunotherapy? Do you have a single answer? Unfortunately, the answer was not a single biomarker. Why is that so? Because as soon as this immunotherapy was available, there were many trials that were done on different cohorts of cancers. And then subsequently, a retrospective analysis was done on three different types of biomarkers. One is the PDL1 expression, and the other two are the genomic signatures. Why do we call it a genomic signature and not a biomarker? Is because this particular alteration or the mutation does not happen at a single location. It happens across the entire human genome in different locations wherever there is a defective region. So that is the reason why you call it as a genomic signature. And today, what we know from this entire understanding since 2011 is that there is no single biomarker for immunotherapy selection. One has to take a multi-marker approach. Among all the three, the MSI-H or the microsatellite instability high patients or deficient mismatch repair as a phenotype stands out to be ranking number one, followed by PDL1 and TMB going hand in hand. There is no specific rule of thumb that if this works, this does not work, and if this works, this does not work. And TILs are, kind of an ancillary information that supports these findings. So, general rule of thumb is with high TMB, the essentially number of mutations per megabase of the genome, you are going to have a good response to therapy. Your benefit, or rather the immune response, is a response to immune checkpoint inhibitor is going to be good. Now, where is the cutoff? How do you define it? And what are the studies that have proved that initially that this is how it has to be calculated? The very first study, or one of the landmark studies, was done on whole exome sequencing approach, wherein one tries to look at all the somatic mutations, subtracting the germline mutations, and then dividing them with the size of the tumor panel, which is the exome here. Then later came another study from Foundation 1, where they had a panel which was able to derive the TMB score, very score to a whole exome sequencing. And then came MSK impact panel. This is also again, they did the same exercise back, and everyone who developed a panel across the globe, they all went and benchmarked with whole exome sequencing as a global standard, because that is your functional footprint of your human cell. And if there is a mutation, if ever there is a mutation that is going to transform to a protein, which is an oncogenic protein, it should be part of the genes which are translating to proteins as part of the whole exome. And that's why it remains one of the gold standards or benchmarks to look for. And everywhere we see that it's beyond this foundation one and MC. In India, we have multiple laboratories who have developed their own panels, and whenever it is developed, the TMB, whenever it is calculated, it is either calculated by a robust study, retrospectively correlating with the whole exome sequencing data, or adapted from one of these panels or one of these studies, which has proved by the manufacturer that it meets the requirements of whole exome sequencing data. And then we had another independent body called Friends in Cancer Research. They said, okay, we will remain a third party. You can submit your data. We will compare with the tumor exome BAM, and then we will certify you whether your TMB matches to the global TMB cutoff. But this work is still in progress, although there has been a success. It has not been as well adapted as a BCR-ABL IS in every diagnostic lab which does genetic testing across the world. Maybe cost is one of the reasons, and as well as accessibility and connectivity. Coming to mismatch repair deficiency, we have 4 important genes which are part of the mismatch repair group, as we all know. And what essentially happens in a mismatch repair process, or rather when there is a defective mismatch repair? The human genome is essentially the human DNA is made of 4 alphabets, as we all know, A, T, G, C. There are certain regions in the genome where you have repeat regions. For example, this place, the repeat region has 6 times CA being repeated. Like this, you may have an AT, you may have an ATG being repeated. So the repeats can be single repeat, double repeats, penta repeats, any type of repeat. Whenever there is a repeat, and if one of these mismatch repair processes are not working efficiently, what happens is that there is an error. What essentially the error is, the DNA polymerase, when it tries to copy the parent mother cell to 2 daughter cells, it slips, and either it makes, instead of 6 copies of CA, it makes 5 repeats of CA, or at times in this process it may have 7 repeats. Whenever such an error happens, there is a alert message that goes to the MMR system, and then they come back and they repair this error, provided this entire group of proteins, which are essentially dimeric receptors, heterodimers, MLH1 and PMS2 together forms a dimeric receptor. MSH2 and MSH6 forms another dimeric receptor on the nuclear membrane. So if these guys were intact, this mutation gets repaired, and then the cell is back to normal. If they find that they are unable to repair, they will go and signal, please take this cell to apoptosis or senescence, but it will not be part of any functional activity. However, when one of these proteins are mutated, they will fail to present themselves as a dimeric receptor on the nuclear membrane surface. That is why if there is a loss of MLH1, there is also a concomitant loss of PMS2. Similarly, if there is a loss of MSH2, there is a concomitant loss of MSH6. Because if one protein fails to present itself on the surface, naturally it cannot dimerase. So in such a scenario, what essentially
Dr. Vidya?
happens? Yes.
Dr. Vidya, can you just repeat this very important slide for all the oncologists?
Yes, sir.
Can you repeat again the whole slide so that everybody understands? Very, very important what you are telling to us. I think it will be nice again once more if you can repeat at the end. Okay.
You want me to
Or
do it
summary
right
of
away,
the
sir?
slides
Or
alone again. Yeah,
now?
yeah, you can do it right away also. Again,
Yeah.
once more, can you just, what's
Yes.
happening in normal cells? What is happening in abnormal cells? Why MSH2 and MSH6 goes together, and why MLH1 and PMS2 goes
go
together? So this particular concept, if you can augment again for all the audience, it will be great.
Yes, sir, definitely. So what are microsatellites? Microsatellites are repeat regions which are present in the human genome. So human DNA is made of 4 building blocks. We all know A2Z. So what happens when you keep reading the DNA, human DNA, there is a mixed representation of A, T, G, and C across the genome. However, in certain regions of the genome, these are called low complexity regions, where you will start seeing repeats like this. CA, 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 CA. This is nature made. It's a nature made, and there is a reason why nature has made it like that. Probably it wants a protein to have this kind of a group of amino acids. Okay, when such a thing happens, one has to look at it very carefully. These are called microsatellite regions. So the string which is put up below here is the complementary strand of what you are seeing above there. So when a normal mother cell tries to divide and make 2 daughter cells, what should happen? The 6 repeats should come to both the daughters. So when it is trying to make 2 copies, the polymerase at times can slip and miss 2 steps. That is, one CA gets dropped, and instead of making 6 times, it might make 5 times. At times, it will start working so fast, it's a molecular motor. Constantly, it is like building a house. Bricks are being put top of each other, there is a cement in between, and then keep on adding bricks. While doing this super fast process, which happens in nanoseconds and femtoseconds in human scale, at times the enzyme slips, or at times the enzyme overacts. So instead of making 6 copies, 6 repeats, it might make 7 repeats. So when it makes 7 repeats, this particular group of proteins, mismatch repair proteins, their job is to identify wherever this particular polymerase is making a mistake when the mother becomes 2 daughters. Then it goes and it identifies whether it is a 5 times or a 7 times or 8 times. Anything which is other than 6 times, it will identify the daughter cell. Okay, this is a defective daughter cell because it is not identical to the mother. So let me kill this, or let me repair the daughter's DNA. So when it is trying to do that, it essentially edits and removes the 2 CAs if it is more, or tries to add. This is what happens, and the daughter becomes a normal daughter if you have a proficient MMR system. That means to say, all these 4 proteins are functionally working to the 200% capacity. Where are these 4 proteins located? MLH1, MSH2, MSH6, and PMS2. These are nuclear membrane proteins. In the nucleus, on the surface of the nucleus, MLH1 heterodimerizes with PMS2. Just like we all know in the lung cancer, EGFR has a dimeric receptor, HER2 is a dimeric receptor. Like that, these are homodimers. Here, these are heterodimers because MLH1 is a different protein, PMS2 is a different protein. But they both should come and sit together, and MSH2 and MSH6 should come and sit together to make this a functional process for the repair to happen. So if you see a standard IHC slide, if you see the expression for all the 4 proteins, that is when you call it proficient mismatch repair, and you should see this in WBCs as well as on the tumor cells. However, suppose one of these proteins, if it is a PMS2 or an MLH1 or MSH2, any one of these proteins has got a pathogenic mutation because of which they are no longer able to present themselves on the surface of the nuclear membrane, what will happen? Its partner, suppose PMS2 is mutated, then no longer it is present on the surface. So partner cannot form a dimer with PMS2. Partner is also not present on the nuclear surface. That means to say, whenever there is a PMS2 loss, there is also an MLH1 loss. And you have to see this happening on the tumor cell surface. And similarly, if there is an MSH2 or an MSH6 mutation, one of these proteins, whichever got mutated, is unable to present on the surface of the nuclear membrane. Automatically, it cannot form a dimer on the surface. Therefore, the MSH6 also cannot be presented on the surface. That is when these two proteins are lost. That is what you see in a classical deficient mismatch repair process. And when such a phenomenon exists, what is happening? This particular 7 repeat or 5 repeat is no longer recognized as an error inside the daughter cell, and the error remains. Now, the daughter cell immediately does not get transformed into a cancer cell. It waits because it's just one error. It has to accumulate more errors in order to transform itself to a cancer cell. So, it may happen in the lifetime of the daughter cell, or if the daughter cell ages and then it goes to senescence. Many times what happens, this error is recognized by our repair system and it gets damaged. If the repair system is not intact and it is going for a toss, that is what happens. If it is a germ line, you will see this happening in almost every tumor cell that is present in your slide. Essentially, if it is a somatic one, you will see malignant cells, but not all malignant cells will have this kind of a feature, because in somatic, this particular error is not happening in every tumor cell. It is happening in only a group of tumor cells. This is a very important distinction between somatic DMMR and germline DMMR, which is a very important feature which we all should understand. If it is a germline DMMR, we know for sure that there is a high risk of predisposition and there will be a strong family history, and that goes on. So, so far so good. Any further questions? Now, what type of mutations? These mutations can be point mutations, which are present in the functional domain of these 4 proteins. What are these 4 proteins? Again, these are DNA binding proteins. So, essentially, the functional domain, which has amino acids where you have a critical amino acid that is responsible to bind to the DNA, or a group of amino acids, if they get inserted or deleted by a foreign sequence or deleted, then they lose their function. That is one way nature makes these mutations. The other way the nature makes these mutations is, all of us get 2 copies of these 4 genes from our parents. One of the copy might be having a terminating codon right in the middle of the protein, so that the functional domain is chopped abruptly. That means you cannot have a functional protein produced by one copy, and the other copy is there to actually take care of the growth of the fetus and then subsequently an adult, and then maybe in the 3rd or the 4th decade or the early 3rd decade, the cancer onset happens because it's been constantly burdened by a single allele, and that's what we are seeing. How mechanistically it happens again will vary between the 4 genes that we are looking at. Does this explain the concept of mismatch repair am I clear in the best of my understanding? I tried to present this. Sir?
Yes, Vidya. Yeah, it is very clear actually. Nicely you said. Only thing that nuclear protein you said, when MSH2 is lost, then dimerization won't happen. So MSH6
6 is
also
also
won't.
not presented.
But what will happen at the gene level? If gene level one is lost, what will happen to the another gene that?
So gene level one is lost in the sense, what is exactly? It is a mutation. The DNA still is there in the chromosome. Everything at the end of the day, what we have to understand is phenotype only. If the phenotype is the one, so when you say lost, it is not the entire copy. So entire copy loss is also a phenomena. However, the other copy is there with which it will try to survive. If the other copy also develops a mutation, then that is when the cancer onset happens. So even
Okay,
inside the
thank you.
nucleus, this protein, if ever it is produced also, it will not be produced as a functionally active protein. It will remain non-functional.
Okay, okay.
It has
Yeah,
to present on
we
the
understand.
nucleus surface to work as a DMMR, to work as an MMR, PMMR marker.
Okay. Thank you. If any of any others have doubts, probably this is one of the important slide what she has put. She has explained so clearly. I haven't heard anybody explaining so beautifully. Thank you. If any of you have any doubts, probably this time we can stop and ask doubts. Otherwise, we can proceed.
Hi, ma'am.
Yeah.
Dr. Narendran here.
Hmm.
Just wanted to know, there is a concept of like MLH methylation is there.
Yes.
There will be isolated loss of MLH1. There will be no
Yeah.
loss of PMS2. How that mechanism works? Because if
There also, see, when there is a promoter methylation in the MLH1, it is a somatic alteration, right? It happens as a sporadic event in the tumor cell. When that happens, there also at the IHC level, it will be a loss of MLH1 and PMS2 together.
Okay.
Yeah. So because they both have to dimerase to function. If MLH1 is not produced, only PMS2 is produced. In some scenarios, you might see a PMS2 expression in IHC. However, recently we do come across such cases in our routine diagnostics where although it is like a pathogenic alteration between the partners, one of the partners shows up on the IHC there. Then the pathologist is also sometimes very clueless ki isolated PMS2 expression is observed. Kindly correlate clinically with any available clinical data to understand the role of this phenotype being actually mismatch repair deficient or not. So when we look into literature, these are very scanty cases, but they are there and they are a good representation, not just for MLH1 loss where PMS2 isolatedly it is expressed. It can happen with MSH2 and MSH6 scenario also. However, the case studies where people have tried to use immunotherapy in such patients has not been a very outstanding benefit like what you would see in a classical mismatch repair deficient system. It is
So
like
what in our clinical practice they say is that if MLH1 isolated loss is there, so you go and check for the BRAF mutation and then methylation. So if both the MLH1 and PMS2 loss is there, we don't check for the BRAF or methylation.
Yeah, that is the reason is when there is an isolated or when there is a somatic mutation. In other words, it can be a promoter methylation of MLH1 because of which MLH1 gets silenced, or there can be a terminating mutation or a pathogenic alteration in MLH1 because of which MLH1 function, native function of MLH1 is lost. You have to do a reflect testing on BRAF, particularly if it is if the patient does not have any strong family history, because this is like a kind of a what you call a clinical phenotype demonstration which has been well documented. If you have a sporadic MLH1 loss, there is a high possibility that BRAF will remain positive. So these are like they go hand in hand. That's the reason why it has been
Madam, sorry to disturb. Just one request. Can we have the questions at the end of both sessions, because we have the next session at 6:45, and
yes.
we can have the questions at the end.
Sure,
Perfect.
sure, sir.
Okay.
Please proceed.
Thanks, Suresh.
Yeah.
Yes. Proceed, ma'am. Proceed, ma'am.
Yeah. So, but that question, I recently come across many cases like this. Isolated marker loss in any one of these four. It is a dicey situation because you cannot stamp it as a deficient MMR. And we actually gave some evidences to one of the physician because he was kind of dodging. Should I consider it as a MMR proficient or deficient? Okay, so we do come across here and there few cases like this, and the answer is it is going case to case basis, and by and large, immunotherapy has not been very effective in such patients. That's the evidence we have so far. So this
Thank you.
is one of the landmark studies. In the interest of time, I will not go into the details of the study, where the very first time mismatch repair high or mismatch repair MSI high or mismatch repair deficient is a tumor where biomarker is considered as a genomic signature, where you can see an excellent response with pembrolizumab back in the year 2017. This is the journey of MSI high being a FDA approved biomarker starting from 2017, and where we are in different cancers and across solid tumors with some of the important studies that has been put up here. And moving to the next biomarker, whether it is a TMB or an MSI, how did it evolve from the concept of immunotherapy per se? This is the slide there, and I essentially put up this slide to say the very first time when the approval came for tumor mutation burden being high is in the year 2020, keynote 158. And I'm just shifting gears here just to bring a new concept here on tumor mutation burden. Well, in the year 2020, FDA approval came based on whole exome sequencing as well as complemented with Foundation One's panel, where it was approved that there is a global TMB cutoff of 10, which is very important to consider before looking for an immunotherapy based on TMB as a biomarker. So afterwards, there were many trials and many studies, and it took 2 years for few people to come back and present their work. And there is a new concept here. Do we need to look at the ancestry before we call for a threshold or a cutoff in tumor mutation burden in order to see good clinical response in outcomes in immune checkpoint inhibitor space? So this study included a retrospective analysis of Caucasians as well as Asians and Africans. And in the interest of time, I just want you to look into an important factor that has been mentioned here. Among the patients with non-small cell lung cancer treated with ICI, those who were misclassified as TMB high on tumor-only panels did not show improved outcome, and these were significantly from European ancestries and they were subcategorized between responder versus non-responder. Responders were more from the European ancestry as compared to the non-European ancestry. And then they also showed that the TMB estimates were overestimated or substantially overclassified as TMB high when you do not do a germline subtraction, which is very important, and it was more pronounced in Asian and African ancestry. In
5 minutes are left.
short,
Sorry to
yes.
interrupt. Yes.
Okay, sir. In short, what we want to say here is one has to, when you want to establish a TMB pipeline, it is better you validate the pipeline with a tumor normal subtraction on a given panel. So, and following that, one also has to look at are you taking the fact of ancestry also into consideration. So here is one of the studies, first of its kind from our lab, where we looked at 1,200 plus patients across 30 different cancer types. We first established this in whole exome, and then we developed our own pipeline. The concept here is when you try to arrange patients, any given cancer in the increasing order of the TMB, the highest TMB containing patients fall in the 9th or 90th percentile, which essentially indicates a high neoantigen load and high efficacy to immune checkpoint inhibitors. So I'm not going into, this is just a high level information. What we saw here was, in general, the Asian population, the trend of TMB scores was higher as compared to Caucasian population when we compared it with the Cancer Genome Atlas data, which is predominantly Caucasian, and we call this as a public cohort. And in 4 different cancers, sarcoma, ovarian, head and neck, and breast, we saw that the TMB median TMB thresholds in the Asian data was very different from the Caucasian data, unlike in lung and colorectal. So this is the data distribution. So before you get into any tumor-only sequencing, first you validate with tumor and normal and try to see if you are able to do tumor-only and get the same value. This is our correlation graph. And this is the TMB cutoff calculation, where you see between the 4baseCare cohort, that is a cohort of 1,200 patients, versus the TCGA cohort. You see here the 90th percentile. In the TCGA cohort, the highest score ranks anywhere around 12.8. That is why in the Foundation 1 data, it was coming somewhere around 10 and above. Whereas in the Asian data, somewhere around in 20. And when you go cancer-wise, you definitely see that not all cancers have the same kind of distribution of TMB ranges. Different cancers have different TMB score ranges. See, Merkel cell carcinoma, numbers are small, but you see very different range. When you come to pancreas, again, very different range. So when it comes to brain, it is very different. Cervix, very different. So every cancer has a different TMB distribution. That's the take home. That means to say every cancer's TMB threshold in the 90th percentile to call it a high neoantigen load is again different. This is the graph that shows essentially that the median scores are different across different cancers. And now there are few studies which says that though we have 3 different types of biomarkers, do we always rely on only one biomarker, or we need to rely on TMB also, or do we need to rely on PDL1 also? Some of these studies, this is on the endometrium, C endometrium, 4.5% patients who were not MSI high. Okay, in other words, they were mismatch repair proficient, were TMB high. This indicates that if you just went by a mismatch repair screening protocol, you are missing close to 5% patients. On the other hand, if you went by TMB as a criteria, you are missing 4% of patients who could have been DMA mismatch repair. So these are 2 important studies that brings out this. So in the TMB high group, 6 out of 31 did have mismatch repair deficient phenotype. Like this, we have different studies where they have time and again proved that you cannot rely only on a single biomarker. You might want to look at both MMR and TMB before taking a call. Similarly, in colorectal cancer, there are data which shows that you need to look at multiple biomarkers before you get into better classification, whether stratification of patients. In head and neck also, although this particular tumor does not require to look at any biomarker, nowadays you give nivolumab, but there are studies way back where they have shown a distribution of TMB and how it has correlated to overall survival when they were treated with immune checkpoint inhibitors. This is one study where mismatch repair proficient tumor but high TMB and no PDL1 expression, excellent response to therapy.
I think, ma'am, we need to wind up the session.
Another case of carcinoma parotid gland. Here is an important message that comes from cancer-specific TMB and PDL1 expression. See a parotid gland. As we know, we often look at HER2, but this is back in the year 2020, 2019. This patient presented with a mass, and then mass, it's a slow growing mass, and it was there for a very long time, but then it subsequently became malignant. And then this is imaging, and then when you look at the histopathology, it was confirmed it's mucin producing adenocarcinoma. And then the patient opted for a surgery. Following surgery, the patient was and then based on the surgery, and the classification turns out to be like this: T4 and 3B and PMX. And then patient was subsequently suggested to go for a radiotherapy, but he refused to do so. And then he was just on a follow-up with nab-paclitaxel. Then he developed recurrence, local regional recurrence in the April 2021, between close to 6-7 months. And then at that point of time, the doctor advised a CGP. And in parallelly, the CGP results came, and then it suggested there is a high PD-L1 expression. TMB was 5.67, purely based on PD-L1 in the year 2021. Should we go ahead with immunotherapy or not? Because that was the only choice left. And then when you looked at the TMB score, we realized that it falls in the 95th percentile for this particular patient, indicating a possibility of high neoantigen load. And then the follow-up, the patient, was given immunotherapy post CGP. And this is where the patient is and is doing fine even today. Thank you for your time.
Thank you, madam. It was an excellent presentation, and you started from hallmark of cancers to pathogenetic drugs and emerging biomarker. I think